Okay. Well, welcome everyone to today's session of the Soil and Nutrition Conference. Today we're welcoming Professor James White to speak to us around the topic of rhizophagy, how plants and microbes work together to facilitate the uptake of nutrients in the soil, and how those microbes cycle from the soil to the plants, how that whole cycle works. James, it's such an honor to be able to introduce you and to listen to your presentation today. I've been just really enthralled with all of the information you've been sharing since 2018, since I heard your first talk with John Kempf, and I believe any available talk, presentation, or podcast or anything you've put out, I've tried to glean more information from. So I'm so looking forward to today, and I will just give you the platform and sit back and take notes.
Thank you, Faith. That was a really nice introduction. Thank you very much. What I'm gonna do now is I'm gonna try to share the screen. Okay, good. So as Faith mentioned, I'm going to be talking about the rhizophagy cycle, and this is actually a process where plants are managing soil microbes. They attract soil microbes to their roots, and they internalize them into the roots, and they extract nutrients from them using reactive oxygen in the roots. And I'm going to talk about that process, but I'm also going to talk about endophytes in general and another process that's happening. Endophytes or plants are getting these microbes out of the soil, and then they become endophytes in plants, and then plants use them in various ways. So the first source for all of these microbes that go into plants is the soil. These are soil microbes. That's why it's important to maintain healthy soils so that your plants can get these microbes that it needs to develop. And I'll show you how this whole process works in the roots, the rhizophagy cycle, but I'll also show you how plants can obtain nutrients from these endophytes that are in the leaves, also in trichomes in places that you don't think of, or essentially leaf hairs, trichomes. Endophytes are any microbes that go into plant. The only important thing is they don't cause disease. And you may have heard of endophytes with turf grasses. You've got a fungal endophyte in certain grasses or forage grasses. And it's inside the tissues. You don't see it unless you have a microscope. And this actually shows an image of one of these fungal endophytes in a grass plant. Endo, meaning inside, phyte, meaning plant. So any microbe really that goes inside a plant is an endophyte. Fungi, bacterial, algal. Okay, but plants, all plants have endophytes. There's not a single plant that doesn't have endophytes. It's not just in grasses. They're in every single plant that you can think of. And these endophytes will grow, particularly what's interesting to us is the ones that go inside the cells, inside the plant cells. So the plants will actually take these microbes and put into their cells. And you can see them in root hairs, you can see them in other root cells, but you can see them also on leaves and in the leaf trichomes. But we're going to talk about first this rhizophagy cycle and what's happening in the soil. And to see these endophytes, you have to have a microscope. And beyond that, you also need to have certain stains. And I've listed some of these stains down below. DAB, NBT, ammonium molybdate, aniline blue, acidified diphenylamine. Okay, those are some of the stains that we use to visualize endophytes in plants. And this first one, DAB, is the main one. And you'll see most of the images we've done with that one. Okay, here's a typical plant, cactus. This is a cactus that occurs on the desert island of Bonaire in the Dutch Antilles. And that's one good thing about doing field work is I get to go to some interesting places to do some work. And in fact, when you love what you're doing, it's indistinguishable to tell work from vacation. And so this work with on this bonaire really, you can see that. Okay, these cacti are huge. They're all over the island, but they have fruits way up. You see the fruits, and if you take those seeds out like that and then germinate those seeds, you get a seedling that looks like this. And this one actually was stained for hydrogen peroxide. It's a form of reactive oxygen with this DAB, and you can see the kind of reddish brownish around the roots. That's because wherever the microbes are, there's a lot of hydrogen peroxide because of the way the plant interacts with that microbe. And so then, because we use these hydrogen peroxide stains, we can see where the hydrogen peroxide is around these microbes, and we can see the microbes better. This is a close-up of some of those hairs, and you can see here, these are the root hairs, and you can see little dots in there. Those are loaded with bacteria, with dots, all in there, all in those hairs. These hairs become filled with microbes. Here's a hair, here's another hair. You can see the arrows now, the lower arrow, you can see kind of two little dots there. Those are where those bacteria have divided. So these bacteria are actively growing inside the plant. I like to think that the plant is replicating these microbes because the plant is actively involved in this process of cultivation of these microbes. And I'll show you what I mean by that in the rest of the process. The plant is very much in control of what it's doing with these microbes. This is not a pathogenicity. Plants take these microbes in from the soil. They don't take all microbes in, but they take some of them in from the soil, and then they process them inside the root cells in this way. And all plants have endophytes. And generally, it's a community of endophytes in plants. So they have selected, plants select the community that helps them to grow the best. That's how we think about this process, obtaining the best nutrients and are the healthiest under the circumstances. Okay, what about crop plants? Okay, that cactus wasn't a crop, it's wild. Crop plants, all of them harbor endophytes. Here's one on one of my graduate students, April Michi, is trying to work on endophytes in cannabis. Of course, it's not quite legal here, so it's kind of hard to work on this right now. But we do have some people who are approved, so we can do a little bit of work on it. But if you take the seed of cannabis, it naturally carries endophytes on it. And in the seed, and you germinate that, take a look at it, you get this. These are the root hairs off the root. And this is now stained for hydrogen peroxide. You can see the little reddish brown in there. Those are bacteria inside that root hair. Look at the root hair to the right. You can see the large spherical structures and smaller spherical structures in there. These are the bacteria that have been internalized into those root cells. This is very much an active process of getting these microbes out of the soil that plants are engaged in. This is how plants get their nutrients naturally, rather than putting nitrogen, for example, or inorganic forms of nitrogen or other kinds of nutrients onto the soils. So this is how plants naturally get their nutrients. They get it from microbes. And that's the importance of having a living, healthy soil. Here's another plant that has endophytes. This is tomato. You think of you cut open a tomato, you think, oh, those are 100% pure, clean, sterile inside. A lot of people thought that. But if you take these seeds out and germinate them, and you can actually isolate endophytes out of the seedlings, even if you sterilized them a good amount. This is one of the endophytes that come out. This is one called Micrococcus, and it produces these tetrads, or little clusters of 4 bacteria together. And we like this one because we could take this one, kind of like our guinea pig, microbe guinea pig, because we could take it, we could put it in a plant, and it'll retain its microbial, its tetrad forms until the superoxide oxidizes those walls off, and then they'll form these little protoplasts. So we can track it in the plant at the various stages and see the transformations that happen with this microbe. And so this is actually tomato root now. You can see the root hairs over here to the left, and you see the tip over here to the right. And you see this orange area this large orange area, microbial entry zone, the bacteria are going in there. They're going in there and secreting ethylene. And we can see that ethylene coming up as this purple orange kind of stain. Well, they also go at the tip, and there's some purple out there. They're also entering right there at the tip. So they're entering two places in these roots. They're coming from the soil, being attracted to this area, and they're going into the cells there. And I'll show you more of that. So this is that Micrococcus, that tetrad, and you can see this was, in this case, we put it inside a Rumex crispus, a curly dock, and you can see the cells here, and you can see the big cells, those are the root cells, and you can see the little blue particle cells, those are the bacteria in their tetrad shapes. And so they have their walls on them, cell walls on them, and then they form in these tetrads, and you can see them. So when they first go into the root, at the root tip, they will have their walls on them. After that, the plant will detect their presence and secrete superoxide on them, which will oxidize off the cell walls and start to extract nutrients from the microbes. So the plant is extracting nutrients from microbes using superoxide that it produces in response to presence of the endophyte. I'll go into more of that. You can see the bacteria in the cells here. This is just so that you can see for yourself these tetrad forms, little square tetrad forms of bacteria that have been internalized into the root cells. Okay, so I just want you to have a good, clear image of that, so you see the bacteria going in. Well, okay, these microbes are from the soil, so they all come at some point from the soil that the roots take out of the soil. But the best microbes the plant will put on its seeds, and you'll see them on seeds, and also use them in other parts of the plant. Use them, for example, in the trichomes that we'll talk about later. But right now we're talking about Rhizophagy cycle. And this is not something that we invented in my lab. We're on a separate, really a track working on these plant microbes, bacterial endophytes, when the paper came out where someone actually described these intracellular endophytes. And this was actually, the discoverers were a group in Queensland, Australia. This is one of them, Channy Pongfu, Dr. Pongfu. This is their work. What they did is they took yeast, they took bacteria, they labeled them with GFP so they could track them in plants, and they fed them tomato, and they fed them to Arabidopsis, and they tracked them in the tissues to see where they were. And what they found is, if you look here to the left image E over here, all the way at the bottom, you see a root hair, and you see those little red or little green dots in there. Those are yeasts that have been stained with GFP. That was their stain. They tagged it with green fluorescent protein, so it would fluoresce when they looked at it. And so now you can see that those bacteria that they tagged went inside those hairs. You see them in these other pictures as well, at B and D, all those little dots in there. All the bacteria that they labeled and fed into tomato or Arabidopsis. I'm not sure which one that is, but it went into both. These investigators later called this Rhizophagy for rhizo root, phagy for eating, root eating. And we, in my lab, we expanded on this, try to figure out what was going on here, and we filled a bunch of details in. And what we figured out is that it's actually kind of cyclic, this process, and we called it the Rhizophagy cycle. And that is that what's happening is microbes are in the soil are being attracted to the roots because the roots are secreting exudates. These are sugars and organic acids, and in some cases amino acids, so that the microbes are able to find the plant. And then once they find the plant, they are internalized into root cells at the root tip, at the epidermis area. And we don't exactly know how that internalization occurs. I mean, this is simply an area of unknown there. We do know that they go in because we can see them going in. We can track them in. We can see what happens. And right now we're left to speculate about, what is the process? For example, is it endocytosis? Is it phagocytosis, where the plant cell just engulfs the microbe and takes it in? Or is it some other process? For example, where the microbe uses its own digestive enzymes to get into the cell wall, and then once in there, they're trapped. We don't exactly know. We do know once they get in, though, they can't get out. And what happens once they're inside these cells is they're hit with superoxide. And that superoxide will digest the cell walls off of the microbes. So losing their cell walls, whatever nutrients were on those cell walls, like proteins, for example, go straight to the plant. They can absorb that. And any other metals that were on those cell walls of the microbes. And also some of these microbes are completely degraded. So whatever nutrients they had in them, for example, potassium, which may have been in the cytoplasm, then goes straight into the plant. So the plant can get a lot of nutrients from these microbes in the Rhizophagy cycle. And they do that. This happens all the time. All plants are doing this. And we can make it where it doesn't work as well by putting nitrogen on it, which paralyzes the process, or we could favor the process in some other ways, putting up other kind of organic nutrients or other nutrients that plants can get to. So what happens is the microbes are extracted of nutrients using superoxide, then they accumulate, surviving microbes will accumulate in these root hair initials. And then they will trigger root hairs to grow. We know because without microbes there, we have no root hair elongation. Those microbes trigger root hair elongation. And then as the hairs elongate, those microbes are ejected out of the tips of the hairs back into the soil. And you can see these arrows coming out of my hairs here. That's every time that a hair grows, it ejects some of those microbes. They go to the soil. They then reform their cell walls and their cell shapes and go out acquiring nutrients later to be attracted back to the plant. We don't know a lot of details about this. You know, which particular microbes are involved in giving nutrients, which ones are fully degraded, we don't know. Which ones are problematic if they go into the cells, because there are some that could be problematic and the plant doesn't manage them as well. We've seen some of those experiments, but so there's a whole range of possibilities here for these microbes. And what we do know is that the Rhizophagy cycle happens, microbes go in it, and plants get nutrients as a result of it. Okay. Plants with more root tips would get more nutrients from Rhizophagy cycle. And the reason for that is that the Rhizophagy cycle is a phenomenon of root tips. And the more root tips you have, the more nutrients plants can get from this process. And this is a grass root. Grasses in particular have a lot of root tips, a lot of root branching. And on all these many tips, Rhizophagy cycle would be happening. So a grass plant like corn could be doing a lot of Rhizophagy cycle in those roots, would be doing a lot. Okay, so this shows a grass root. This is actually a Phragmites, one of our experimental plants that we work on. It's a root. You can see this is the exudate zone. That's blue cloud out here. These are all the microbes that these roots are attracting to them. And they're absorbing them into the cells right at this, right at the epidermal layer at the tip of that root. Excuse me. This is what they look like inside the cell. Now, this is a root epidermis cell. This is of Phragmites again. And you can see these black arrows, they're showing the bacteria inside. And you can see they're replicating in there. They are replicating inside that cell. The smallest ones are the youngest ones. The oldest ones are the biggest ones. And I used two stains here. One is the brown for superoxide, or rather for hydrogen peroxide. The other one is aniline blue and blue. And if you notice this lower arrow, that smallest cell is blue. That's because of protein. That's a protein stain, aniline blue. And so where there's more protein in those cells, you see those staining bluer. So what's happening is these cells, as they age, they're being penetrated by this superoxide, which goes right through the membrane and will break down proteins inside those cells. So inside those bacterial cells. So there's proteins being degraded. And so that's one source of nitrogen for the plant is degradation or oxidation of these microbial proteins that would then form, or even larger molecules. We know that plants can absorb breakdown products from proteins and from other kind of DNA molecules and so forth. So larger molecules could be absorbed using this same system. So these microbes, actually, they're not in the cell proper, that is, they're not in the cytoplasm. Instead, these bacteria go underneath the cell wall. This gray area here is the cell wall. And you can see the central area here, I'm showing, that's the cytoplasm of the root cell. They're not actually in that. They're outside that plasma membrane, just underneath the cell wall. So they're in this space called the periplasmic space. So the plant can detect when those microbes are in that periplasmic space. And I'll tell you the take home now is that they can detect them because these microbes are producing ethylene. And so they can detect that ethylene and then they will actually respond by secreting superoxide. And that's just 2, 0, minus. The 0 minus out here, that's the superoxide that's being produced and put on those microbes. And at the same time, the plant is managing these microbes. It's moving them around in the cell, okay, in a process called cyclosis. Cyclosis, C-L-Y-C-L-O-S-I-S, cyclosis. That just means cell movement, and I'll show you how that happens. But it's important because this is how the plant is managing these microbes, keeping them off their toes, mixing their nutrients up, encouraging them to, moving them around like that, encouraging them to divide and so forth, replicating them. So this is very much part of this manipulation process, or farming. I like to call it farming, where plants are farming microbes. This is a farming process. This actually is the enzyme that the plant uses to produce superoxide, and it's one called NADPH oxidase enzyme, or sometimes called NOx enzymes. And if you're familiar with the way aspirins work, they usually work on certain of these reactive oxygen enzymes. So this is one of those that plants have, and this enzyme will take molecular oxygen from the atmosphere, and it will produce superoxide. And then that superoxide will use to get nutrients out of the microbes and to control the microbes inside the cells, inside the root cells. Well, this, because superoxide, I almost forgot what I was going to say. Because superoxide is necessary for Rhizophagy cycle, if plants don't have oxygen in the soil, they don't have good aeration, they cannot use this enzyme to produce superoxide, because it takes molecular oxygen and will convert it into superoxide in this using this enzyme. And so if you have poor aeration in the soil, say you've got a hard pan or some other problem causing flooding, or just non-good airflow, too much lack of oxygen in the soil, plants are not going to grow well, because they need that oxygen in order to produce superoxide, in order to extract nutrients from the microbes. So you right away, you're going to reduce nutrient extraction if you don't have good airflow in the soil. Well, plants will hit those bacteria with superoxide, and that will cause the bacteria that have their walled shapes, like these over here to the left. This is a bacillus rod. See the rod shapes. But once superoxide oxidizes off those cell walls, instead you get these spherical structures shown here to the right. These are the protoplast, bacterial protoplast phases. Fungi will do it too. Certain yeasts will go in, and they also hit with superoxide. They will lose their cell walls as well and form these protoplast phases. In bacteria, they call these L forms. They're just wall-less bacteria. In fungi, they call them, was just there just a second ago. Mycoplasts, I think, or something similar. They're just little naked protoplast fungal phases, and they're basically the same, between fungi. And so fungi can use bacteria to get nutrients. They could also, plants can use bacteria to get nutrients. They can also use fungi. But they're important for more than just obtaining nutrients, because the plant development depends on presence of these microbes in the root cells. And that is, the microbes in the root cells will trigger the gravitropic response. The gravitropic response is when you have roots that go down with gravity. If we take all the microbes off of plants, off of seedlings, the seedling roots stay on the surface, or sometimes they just go in the air. So it's necessary to have the gravitropic response, to have the microbes there. Also, root hair elongation. If you have no microbes on a seedling, you have no root hairs forming. You may have root cells growing, but you don't have bacteria in the root cells in order to trigger root hair elongation. Okay, and so microbes development in plants is closely tied to the presence of microbes in plants. And the reason is, part of the reason is because the microbes, besides providing nutrients, the microbes are also providing growth hormone, ethylene, at least one that we've been experimenting, we found. Ethylene, it's an important growth hormone that triggers elongation of growth in cells. And okay, so this is actually a Bermuda grass in which we removed all the microbes from it. You can see you have a nice root there, but there's no hairs on that root. Okay, when we take in that same experiment, we then take a microbe, an endophyte that we have off of our Phragmites, put it on this Bermuda grass. Now, then you see what happens right away. Now we put in this grass, we put this is a root here. We put the Pseudomonas bacterium on it, and you can see right away. You see hair formation right away, and hairs form right away on this with the bacterium. Okay, this shows a bit later on. You can see all oops, go there. You can see the hairs, all the brown in there. This is stain for hydrogen peroxide. All that brown is bacteria in there. And if you look at the root itself, the little brown dots, those are bacteria inside there. These roots are filled with bacteria. They're filled with bacteria. This shows one of these root hairs. You can see all the little spherical, those are the protoclast phases inside there. That's a pseudomonad. So that's Pseudomonas bacterium, common soil bacterium, common plant bacterium. Okay, tomato seeds have bacteria inside them. And it's actually tricky to kill. They have some on the surface, but they have some inside them. It's tricky to kill them, but you can if you're very careful with antibiotics. This actually shows one of these experiments. This is a tomato with no antibiotic treatment. You can see the seedling there. You can see the hairs coming out. Same age now, but treated with streptomycin. Okay, in the seedling stage, and you see there's no hair formation now. We didn't negatively impact the growth as far as we could tell of the seedling, but we killed the bacteria or inhibited the bacteria, and you have no hair formation, no elongation. Okay, this just shows this. This shows a hair in which we removed all the bacteria. You can see they formed these hair initials called trichoblasts, actually trichoblast. And you can see in there, there's nothing inside there. You don't see any bacteria in there. Whereas over here at A, okay, we have our bacteria still on this tomato, and you can see the little brown dots in there. Those are the bacteria in that trichoblast. And you see as it elongates, you can see the bacteria concentrate at the tip. Now they stay at that tip, and hairs elongate. You can see here a bit older. You can see the microbes over here, all that brown in that tip of that hair. Many bacteria go into a cell, a root cell at the tip, but certain ones are replicated, and those ones that are replicated are produced in massive numbers as they go out of these root cells, root hairs at the tip. And I'll explain that. But the soil essentially, the plant essentially repopulates the soil with these microbes that work with it well in the rhizophagy cycle. So these are the ones that go increase in concentration. And why is there this linkage between growth and hair formation? And my graduate student, this is Ivy Chang, who's working on this, but she's looking at the what is it that, how does the bacteria cause plant root hairs to grow, and what stimulates root growth in general, having to do with these microbes. And here you can see a grass without microbes. You can see there's no hairs on that. This is stained for hydrogen peroxide, but there's no hairs there. And you can see here to the right where she put the microbe on it. This is a Pseudomonad again. She put it on, you can see the hairs right away forming on that. So ethylene is what we found is being produced by these microbes. And all of our data, we did many experiments to support that. Ethylene is also a growth hormone. It's a plant hormone that plants will use to trigger growth and maturation in fruits. But it's also something that these microbes are producing, the ethylene, and they're able to use that, trigger the growth, the roots, plant roots to grow, and then the microbes can get nutrients when those cells are growing. Okay, so we figured out that this is actually the interaction, the chemical interaction. This is one of two important chemical interactions that's happening between the microbes and the plants, intracellular microbes. And this I'll just show you just a few of these, just so you see what I'm talking about. Okay, one reactant is ethylene. The microbes are producing ethylene, and the plant then, in response to that ethylene production, will release sugars to the microbe, but also releases superoxide. And that superoxide then will combine with the ethylene and will form nitrogen peroxide. And so when we stain with that nitrogen peroxide stain, we're able to see the bacteria, because the hydrogen peroxide is all around those bacteria, being the product of that ethylene and superoxide combination. But of course, it's that superoxide that extracts nutrients from those bacteria. So that is, but that's a part of that interaction. They enter in this area, and we'll look at close up of some. Okay, so this is actually from the tip. Here you can see this is one of these root cap cells that have peeled off, and you can see the bacteria in here. This is stained for ethylene now, using an ethylene stain that makes it purple where there's ethylene. You can see the purple around these spherical structures. That's the ethylene being secreted by those microbes. This shows the root itself, and you can see the microbes on the outside. That's where they're penetrating in, right there into the root cells. And this is a close-up now. You can see this is the root cell. You can see the spherical structure inside with this arrow. That's the microbe in there. You can see the purple around it. That's ethylene that the microbe is producing. And these are actually in little pits, so you understand the relationship. This is this microbe is pressed into almost like a foxhole-like depression on the surface of the plasma membrane. So the bacterium is very close to the plasma membrane of the root cell. So they're just right up against one another. The membrane connection. The walls are gone that separated the bacteria and the plant. They have only their membranes now, and they're interacting chemically inside that plant. This shows superoxide. That's that's what the plant is producing to extract nutrients from the microbes, and all this blue actually is the superoxide there. We use NBT to visualize that. This shows the hair, and this is stained for hydrogen peroxide. You can see all the brown around those bacteria in that hair. And this is probably not one species, by the way. This one is from nature, so it's probably multiple species of bacteria in there. There's another reaction that's important, and And it happens to be this reaction here. And in this reaction, what happens is the microbe secretes an antioxidant, but that antioxidant is nitric oxide. It's a potent antioxidant, and it will combine, in order to protect itself, the microbe secretes this nitric oxide, and it will combine with superoxide and will form nitrate. And that nitrate can then be absorbed using nitrate transporters. And in this reaction, what happens is the microbe is essentially providing nitrogen to the plant in order to protect itself from that superoxide. At least that's how we hypothesize this. And we can measure all of these components in plants. And because this is a root hair of cabbage, looks like, and you can see the bacteria there, and you can see the purple around it. That shows the nitrate. That's stained for nitrate. With this nitrate stain that stains purple where there's nitrate. You can see them around all of those, the purple around all of that. So they're all secreting nitrate into the plant. That's how we interpret that. These interactions, when taken together, represent a kind of a trap for these microbes. Again, the plant is in control of this process, because what happens is, if the microbes in here, if they secrete ethylene, they get sugar, this carbon, they get those nutrients. If they stop secreting ethylene, they don't get it. They don't get nutrients, and they starve, and growth ceases, and they can't replicate. So they can't stop that ethylene secretion. In terms of nitrogen, if they stop secreting nitric oxide, what will happen is the superoxide will damage them. That will oxidize the cells, and then they will break down. So they have to keep secreting this nitrogen, this nitric oxide, in order to counteract superoxide, plant-produced superoxide. So this forces them to keep fixing nitrogen if under certain whatever circumstances possible, and then transfer that and use that nitrogen to make nitric oxide. Okay, so you have these loops, and the microbes cannot exit from either of these loops. They're only when they're ejected can they get out of these hairs. And we think that there could be some nitrogen fixation occurring in these hairs. If so, it could be at the sides of the hairs where there's very little reactive oxygen around these hairs, because we've got microbes being replicated around the length of the hair, and also we have less superoxide there, and so that those microbes can potentially fix nitrogen. Okay, so in that process, cyclosis, in the process of rhizophagy cycle, cyclosis is very important. That cytoplasmic strain. This is one of the plants on that desert island of Bonaire, grows in the rocks without really much soil. This is a root hair now, looking at root hair under the microscope. You can see the movement. That's what cyclosis is happening. Actually, if you look below, you'll see one of these root hairs, and you can see the bacteria in it. The brown areas are stained. That's stained for hydrogen peroxide around those bacteria. You can see the bacteria in there, but now the one above shows them living, and they are moving. This is what actually happens. They're constantly moving. The plant is moving them around, replicating them, getting increasing numbers of the microbes, exposing some under certain circumstances to superoxide and getting nutrients out again. Most of the superoxide exposure happens to be at the tips, though, rather than on the sides. Okay, so we'll look at that. Okay, so this is actually showing one of those root hairs again from that sedge. And you can see there's a lot of bacteria here. Those probably originated from one single bacterium. And you can see the biggest are the oldest ones. The biggest ones are probably the oldest ones, and the smallest ones are probably the youngest ones. So you have this replication happening. And when that replication is happening, cells can fix nitrogen and build up their nitrogen contents through nitrogen fixation from the air. but inside the plant, when superoxide really comes down on these microbes, it's going to break down much of that nitrogen that's built up. This shows one of these sedge root hairs again. You can see the whole thing, see what's happening, the bacteria going around in cyclosis. And you see they're accumulating at the tip. They're actually accumulating there. And as those concentration builds up at the tip, these microbes produce more ethylene. And that would then cause a growth spurt to happen. And microbes then, the root hair would grow, and some of those microbes would eject out. I'll show you that. This, by the way, you can actually see these moving in the microscope. So you could look in the reverse of the plate and you can see the cyclosis happening. This is how root hairs really work. It's not the static things that we think of that just absorb chemicals. They're manipulating microbes, they're managing microbes. Okay, so let me see if I can go to the next. Oops. So this is what ejection looks like. This is one of these hairs, and you can see the bacteria outside it, and you can see bacteria inside it. These hairs become fat with bacteria. Fat with bacteria. They're becoming gorged. You can see how fat this is on the end. And some of these bacteria aren't stained. You see some red ones in here, but there are some that didn't take up stain at all, and they're out there. Outside and internally, these hairs are fat with microbes. Literally fat. And the ejection of microbes corresponds exactly to growth spurts in the hairs. And this shows one of the hairs. You can see the microbes out. You can see kind of in transition, bacterial protoplasts that were kind of in that wall that's coming out. Little pores form in the growing wall, and it's through those pores that these microbes get expelled. And this shows actually the way the tip works. This is something we call the cyclosis expansion wave hypothesis for microbe expansion from root hairs. And what you have is this diagram to the left here will show the cyclosis of microbes going around and around. You can see they're being deposited here in this more flexible tip of the hair here, of the wall there. It goes in there, and the tip of the hair itself really is a little needle-shaped structure. So it's skinny little thing inside that. But the microbes accumulate around here, and then at some point, there's an expansion wave. We think when ethylene builds up to the point that it triggers this expansion, this growth wave, happens at the base of the hair, goes to the tip, and essentially squeezes those microbes out of pores in the tip. And I'll show you that. Okay, this shows a hair of a Bermuda grass where we saw those periodic ejections. So at each place here, you would have seen an ejection. And instead, what happened here is some of these microbes were left inside the wall. So they remained on the inside rather than being ejected. So you have those that remain there. Others were ejected. This endophyte comes from another plant. So it wasn't adapted to this Bermuda grass. So that's why it probably doesn't work perfectly when it goes in to Bermuda grass. But this shows these steps in ejection. You can see the red here shows where microbes are producing ethylene and being exposed to superoxide right there. That causes the plants to secrete more nitric oxide, and then that forms nitrate to control some of that superoxide. And then that ethylene that was produced triggers a cell growth event, an elongation. And as that happens, that growth spurt happens, those microbes are shot out. And then the remaining microbes that are in there that were on the sides and were not ejected, then are replicated. And they can be replicated inside during cyclosis when reactive oxygen is minimized, and they can replicate those microbes, and then it's ready again. It builds up and you have another growth spurt. We estimated that is about a 15-second, at least in Bermuda grass, a 15-minute interval, 15 minutes, and then there's another growth spurt, 15 more minutes, another growth spurt, and so forth. But that number is very, highly variable. It's an estimated number. I mean, we could be talking about an hour or 2 hours variability depending on the plant. We really don't know a whole lot about it. This is tomato root hair, and you can see the bacteria from these tomato root hairs actually being ejected through the pores at the tip of that hair. You can see there's some like 3 little pores on number 3 there. Looks like they're going out of those. You can see some over here too. Hard to see the pores in number 2. But this is what this ejection looks like of bacteria coming out of these hairs. So once they're ejected, they would then go out, acquire more nutrients, reform their cell walls, acquire more nutrients, and attract it back to the Rhizophagy cycle. So we did a number of experiments to evaluate what nutrients come with. This is one of them. This is done by a visiting scientist, Dr. Zhao, who took wheat, removed all the microbes off of wheat, and then put back onto the wheat one at a time the microbes, and then measured the nutrients, measured the effect on the plants, and also measured the nutrients that went into plants with and without the microbes. And basically, he cleaned the microbes using Clorox and antibiotic to clean it afterwards. This shows the plants. This is the control, the sterile control, versus the plant in which we replaced bacterial endophyte, happens to be a bacillus. You can see how tiny the roots are in the controls without bacteria, without endophytes, and over here they're huge with endophytes. You can see how the roots are much bigger, the shoots are much bigger. We really see it in root growth when you remove endophytes. So roots really do require these microbes in them in order to grow bigger and better. I mean, that's just like a crude effect. You see it, oh, bigger. Nitrogen, this is terms of nitrogen. Now he took the leaves of the plant and then measured the nitrogen that was in that tissue. And here you see with no bacteria, it came up to this bar, that much. And then with these bacteria, there were 3 different bacteria that he used. And you can see it increased it by about 30%. And that's about like, that appears to be like what the Rhizophagy cycle is worth in terms of nitrogen. It looks like it's about 30% coming into plants from the Rhizophagy cycle, 33%, something like that. It may vary depending on the plant, depending on what the plant is doing. So it's not a hard, fast rule. We've seen it highly variable in other plants. Okay, here is phosphorus. You can see with the bacteria versus without. Looks like about 20% more. And those seem to be about equal. Those different bacteria. Here's with potassium. Look at potassium. That first bacterium, it looks like triple the amount that you had in the no bacterial controls. So that one bacterium really benefited the plant in terms of bacterium. The plant may be, I don't know, there may be an intermediate, a medium, happy medium in terms of breakdown of the microbe and the amount of nutrient that comes out of it and survival and so forth. But it looks like this particular bacterium, however it's working, is bringing a lot of potassium. And potassium you would expect to come out of the cytoplasm because it's one of the solutes in the cytoplasm. So the cytoplasm is full of potassium. So plants could get a lot of potassium from cytoplasm with these microbes. Okay, you look at these others in terms of potassium, and it looks like they're like double the control, but they're still not as good. Take-home message is that every microbe is different in terms of nutrients. They're all different. They affect different nutrients. Most nutrients are impacted, some more than others, but most of them are impacted in the Rhizophagy cycle. Okay, here we see calcium. We see the same deal there. Calcium looks like, any of those bacteria are they're nearly equal in terms of benefits, in terms of calcium, but they benefit. Magnesium, same deal. Zinc, looks like that second one is really good with zinc again. And so that's the Rhizophagy cycle. And Rhizophagy cycle is getting nutrients out of plants, kind of out of soil, out of a bulk kind of soil environment, bringing all those nutrients. But there's other benefits that endophytes have in plants, and this has to do with the nitrogen fixation phenomenon. And this is something new that I have not spoken much about, and it's just a new area for us. We're looking at trichomes, and we hypothesize that these trichomes, well, they all appear to contain endophytic bacteria. We hypothesize that they, in fact, are nitrogen fixation structures. And I'll go and explain the reasons that I think that. But these are trichomes, are plant hairs. All plants have some kinds of trichomes, some kinds of plant hairs, at least most of them do. And they're variable, and people don't really understand what these trichomes have been for. They're not just tools for separating plants taxonomically or little ornamentations on plants, but we maintain they're functional structures that are microbiome related. They function, it appears to us in experiments so far, to provide nitrogen, to get nitrogen out of the air. You know, there are structures on leaves or on fruits, they're sticking in the air. And there's a big need for nitrogen in those structures that bear these trichomes. And so they would seem to be structures that are adapted for, this is what we're working, hypothesis we're working on now. And those two reactions that I described from root hairs, they're very important in leaf hairs and fruit hairs and trichomes. These work in the air, not only in the soil air, but now in the air above the plant. And you can see this is the second interaction, or it's the first interaction, ethylene and superoxide. You could have peroxide. So all of that, we think those interactions are happening. I'll show you the reasons why we think they're happening, because we stain for them and we can see them. Note that the plant, or rather the endophyte, secretes nitric oxide again as the source of nitrogen to protect itself from the plant's superoxide. It's making that nitrogen, that's combining, you're getting nitrate. So those two reactions are important. We've looked at 8 or so, this list, however many, 8 or so species or 9 species of plants that have trichomes in several families, including Asteraceae, including Cannabaceae, the cannabis, right? Beans, Solanaceae, Malvaceae, Scrophulariaceae, a lot of plants. Those trichomes are different kinds. There's dendroid trichomes, there's filamentous trichomes, there's glandular trichomes. Okay, and I'm gonna look at each one of those and just show you what we have on those. But we stained all of these types of trichomes for all of those chemicals that I talked about before that have to do with the two equations in plants. And you can see all these checks show where we did a test and we found that those chemicals are present, like ethylene, for example, in this Ailanthus altissima, this is tree of heaven. You can see that ethylene is produced by 3 different tests. We measured 3 different stains, histochemical tests. Superoxide, you can see superoxide is there, and so forth. You get the point of this table. And this is one of the plants that we looked at that we have a little bit more data on. This is one called Portia tree, Thespacia papulinea. It's a tropical tree. It grows all around beaches and in very non-fertile areas. This is my graduate student, Dr. Evelisa Rizari, who worked on that. And this is one of these developing leaves of Portia tree. You can see the little glandular trichomes. Actually, the top arrow shows a glandular trichome, and I think these glandular trichomes will grow into these bigger ones. Sometimes, at least, they'll grow into the bigger ones. But if you look closely at those, okay, like here, looking at the arrows, this is one of these Portia tree glandular trichomes. You can see the little spherical structures there, and you see the purple around it. The purple is the ethylene being produced inside in various places inside. This is developing here. This is a fully mature Portia tree here. They form these peltate trichomes, little like umbrella-like structures. And you can see the little arms of this here. You can see the little brown in there. Those are the bacteria that are inside these hairs. It's stained for nitric oxide, and it shows the nitric oxide produced by all those bacteria. Here's one of those hairs. You can see where it's collapsed, and you can see the bacteria now coming out. I think this is also nitric oxide around these bacteria coming out of the squished from the cell. So the question is, while we can see these chemical interactions there, does that really mean that these microbes that plants are benefiting by nitrogen that's being fixed by these bacteria that are inside the trichomes? And to answer that, we did some 15 in tracking experiments. This is just one of them. But basically in these experiments, you expose plant to isotopic nitrogen. That's 15N2, basically, because most of the air is comprised of nitrogen that's 14N2 with a lower ratio of 15N isotope in it. So when we beef up the 15N isotope in a chamber with these plants, if they're fixing nitrogen, if they're taking that gas, that 15N2 out of the air, and they're putting it into proteins or ammonia, whatever they're putting it into, we should be able to pick it up by analyzing the plant for 15N isotope and compare that to what we get in the air, and we'll see how much assimilation is happening. So that's what these experiments are about. And so you see these chambers, one with air, one with the N2 in it. You let the plant stay in there for 5, 6 days, 7 days, then take the plants out, dry them up, analyze them for 15N versus 14N ratio, and you get your answer about whether or not nitrogen fixation is happening in those tissues. So this is actually diagrams that shows plantlets, the Portia tree plantlet. And you can see younger leaves right there, the little hearts, and you see the older leaves down below. And if you look at the big numbers, actually, you can see these younger leaves, like the plant 1, you can see there 623.34, and that's a figure delta 15N versus air. It's meaning that's a big number with respect to how much nitrogen 15 was absorbed compared to the roots themselves. If you look down at roots, you see the number is much less than into those, and that goes down as the tissues age. If you look here, get the plant 2, you get 755 at this measurement here. Then you see all the rest of them are down, go down to 2, down to 84 in some of the roots. So you see a lot of fixation is happening on these developing structures, these developing leaves. And in fact, it's in these developing leaves where we have the highest concentration of these glandular trichomes. And so there's a correlation with nitrogen assimilation into plants and presence and density of those trichomes. Here's another plant that we looked at. This is cannabis, hemp. It's legal in New Jersey, but it's still hard to do research on it, believe it or not. But these are and hemp has these glandular trichomes that most of you are probably familiar with. This just shows these developing. Some of these are on a stalk. And the heads are here at the top. So it turns out the microbes are in these heads, by the way. They're in the heads, and I'll show you. This shows these glandular trichomes from the side. You can see the stalk it has. You can also notice the stalk is a little bit green. That's because the stalk has chloroplasts in it. And it's the chloroplasts in this stalk that provide the sugar that drives activities, including nitrogen fixation activities. So we hypothesize inside these glandular trichomes, there's no chlorophyll in these glandular trichome heads. If there were chlorophyll, then we would be generating oxygen, a lot of extra oxygen. That extra oxygen would inhibit nitrogen fixation happening in those. And the chlorophyll below, of course, that's necessary because nitrogen fixation is an energy demanding process and it requires a lot of nutrients. So these stalks have to feed that head in terms of sugars to fix for nitrogen fixation. This is one of these developing glandular trichomes, and you can see the arrows around the periphery say those are the bacteria. This is stained for NBT, superoxide. So the purple around there is superoxide that the plant is producing onto those microbes. And if we look closer at it, you can see here's a bit closer up. And you can see actually now, looking at the left over here, you can see some of these are in the rod forms. These are early on. This is in the rod form. What will happen is the superoxide will oxidize off these bacterial cell walls and they'll form their protoclast phases. But now in trichomes, glandular trichomes, rather than in root hairs like we talked about before. Now the similar process is happening in trichomes. This shows actually one of these glandular trichomes. Looking down at it, there's a leaf below, but this is the periphery of the trichome. You can see the wall of the trichome is out here, and you see all this brown in here. That is all bacteria, bacterial protoplasts inside your trichome. And so that's all area out there that has microbes in it that could be fixing nitrogen. So this would be the nitrogen fixation area out there. Whereas now you look at this, here's in the center, we have our trichome cells, several of them. I heard it's 8. It looks exactly like 8 too. Isn't that 2, 3, 4, 1, 2, 3, 4, look at 8. 8 cells. And you can see where it interfaces with these bacteria now. Now you can see the interface where it's blue. That's this is stained for superoxide. So it's used in that NBT stain that shows blue. And you can see here in this area where it's staining, the cells actually look like they maybe a little bigger, like they're swelling, because that's what you would expect to happen as the cell walls get oxidized off and the cells swell a little bit. But this would be the nitrogen transfer zone right in here, interfacing with that cell, with those bacterial cells. So there's that interface delivers the nutrients. That's how this thing works. We have a model for this. And if you look at the top right, you kind of see that same image that I showed actually below now of that head, but now you see red bacteria drawn in for the little red balls for the bacteria. And you can see the trichome cells in the center, and you see an exchange now. Carbon goes out, that sugar goes out to the microbes, but the microbes push nitrogen, provide nitrogen, put antioxidant nitrogen back in order to counteract that superoxide. And of course, the trichome cells themselves can absorb that nitrate that forms in the reaction between oxide and superoxide. So that's how we think that the glandular trichome is working. There are lots of unanswered questions. For example, we know that these glandular trichomes are sources of chemicals. And in cannabis, for example, the terpenoids, cannabinoids produced inside those glandular trichomes. So do these interactions in the hemp trichome impact on those cannabinoids? And what about hops? Hops also is in that same family. Hops, very similar. And I think I'll talk about that pretty soon. But most likely, those cannabinoids are affected. And the reason for that is that, in fact, the cannabinoids themselves may be what the plant is using as an antioxidant inside those antioxidants inside those trichomes. The terpenoids are antioxidants. They've been shown to have antioxidant effects. I put some articles down there just so you can see that. But there's a lot of data that says that these are THC and similar cannabinoids or other terpenoids inside hairs are potent antioxidants. And in fact, they are rare antioxidants in that they don't have nitrogen in their structure. So this is a kind of antioxidant that does not have a nitrogen cost. A lot of alkaloids that could be produced would require nitrogen, and that would have a cost in terms of nitrogen. But inside these trichomes, the terpenoids are antioxidants that have no nitrogen cost. So it's only photosynthate, no nitrogen. So that'd be ideal for a system that's trying to maximize efficiency of nitrogen extraction from these nitrogen fixing microbes. Okay. Said a mouthful, there's a lot that has to be tested there. But you can see what we're working on, what we're trying to figure out. This is hops, and you can see the hops, these little hops fruits are where all the glandular trichomes are. That's a close-up of a hops glandular trichome. See the arrows there. See the bacteria inside those, just exactly the same way we saw in cannabis. And there you see it. This is stained for nitrate, and you can see on the hops. You can see that it's full of microbes. Hard to see, but you can see all the purple compared to the other epidermal cells. That's all nitrate stain. It's all nitrate. These are filled with organs. Here's a tree of heaven. These are different trichomes. These are they cover the leaves like this, but they're also full of microbes. And this actually shows one of those tree of heaven microbes. You can see some microbes coming out, forming there. They're coming out from the interior of those. There you can see this is a bacillus coming out of this one, but they mostly stay in. So don't get the wrong idea that these are all just to come out for the surface. Mostly these stay in these trichomes and function in the trichomes. Maybe when they get old, they come out, but when it's functional, they so do endophytes affect chemical composition of plants? We did a study on carrot, had a student taking microbes off of different plants, bacterium 1, bacterium 2, bacterium 3 from different, one from celery, one from curcumin, one from parsley, and then put it onto carrot. And then my student, you can see the bacteria now in the carrot root hairs there on the carrot. And there you can see bacteria in just to see what it looks like. And then my student measured the carotenoids in the carrot with and without the various microbes. And you can see the control here. This is with no microbes. You can see this much carotene versus bacterium 1. It's about the same. That bacterium probably didn't have any effect at all. Then you see bacterium 2. You can see it had a big effect. It went inside and greatly increased the alpha carotenes. Bacterium 3, not as much. So one of those was highly effective at increasing. Here's the beta carotenes. It looks very similar. You can see that bacterium 2 increased it. Bacterium 1 just had no effect, and bacterium 3 at minimal. So depending on the endophyte, you can change the chemical components in plants. The endophytes also increase stress tolerance in plants, and they do that because they're having this interaction in the plants, and that's causing plants to upregulate their oxidative resistance because the plant themselves are producing the superoxide. So now it needs to, it needs to be more resistant to that superoxide so that it increases its antioxidants and whatever other mechanisms it has to protect itself from its own oxidants like superoxide that forms inside. Okay, so whenever an endophyte is present, you see this oxidative stress enhancement. Okay, this shows one. This is actually some experiments, an experiment from the experiment that Rusty Rodriguez here did, in which they showed a fungal endophyte made their plants more resistant to salt stress. And you can see here, the ones to the right, they call symbiotic, is where they put a fungal endophyte in their plant, and then they put it in salt soil. And you can see these latter three are the later three. These outer three are the salt ones, and then they compared that to non-symbiotic without bacteria, and you can see that these three salt ones are not doing very well without that endophyte. And that is an oxidative, we think is oxidative protection effect, because that's not, salt is an oxidative stressor. Here's heat stress, same deal. Here they looked at some thermic soils that were really hot, in which the temperature was up to 60 or 70 degrees centigrade. I think it would be very, very hot. Very, very hot, not boiling, but really hot, nearly boiling soils. And the one picture to the left shows the plants with their fungal endophyte, and to the right shows the plants without. And you can see made a big difference having the endophyte present. The endophytes also protect plants from disease. They'll do that a couple of different ways. They'll actually go out from the plant and colonize pathogens and make them to be non-pathogenic. They will also cause the plants to be hardier and less susceptible to disease because of this oxidative stress enhancement that you get. Okay, and this just shows from one experiment, shows actually with English ivy with an endophyte. You see there's no disease there from just the same experiment. You see without the endophyte, you see there's disease all over it. Okay, so this protection has been referred to as like a bodyguard hypothesis. The endophyte is guarding its host, guarding the body of its host using chemicals or whatever means it has to protect its host. This has also been called defensive mutualism concept by Keith Clay with fungal endophytes in particular, the idea that plants are taking in these endophytes as a way to defend themselves from herbivory, for example, or from stressful conditions or whatever the concept would still work there. So you have all of these potential benefits of microbes in plants. I think the take-home message that I want you to have is that plants are taking in soil microbes on a routine basis for nutrients, but also for help them to develop better, to resist stressful conditions, to combat disease. So it would make sense for us to develop an agriculture in which we are trying to support what plants do in terms of how they use microbes, than work against the system by putting on, for example, inorganic forms of nitrogen or something that we know will paralyze the system, or surface disinfect seeds. We know that seeds carry microbes on, important microbes for the plant. So, we should be trying to support plant activities with microbes as opposed to working against it. So these are some papers that we did that are open access, and they have bits of the science in it, and that they're all free. You can but they'll filled in more details than you ever wanted to know. The trichome stuff is not in it, so everything you heard today about the trichomes is brand new. Not very many people have heard it. It's an active research project we're working on, part of my April Michi's PhD project that she's been working on for, believe it or not, for many, many years now. So I think I'm good for any questions.
Oh, that was wonderful, James. Thanks so much.
Thank you.
The new information about the trichomes, that's so fun to wrap our heads around and to really think about how complete the microbiome of a plant is, and how how everything we do as growers really impacts that. So it's really, really great to have you bring that new information to us. We do have some questions, and so I'll go through those. And please, everyone, if you have more questions, just pop them in the Q&A. And then I've got some questions too. I know we'll run up against time, but we'll just see what we can do. Okay, so Roger Weiss asks, what is the frequency of rhizophagy versus the more common rhizosphere well-described relationship where plants attract specific microbes with root exudates and the microbes that produce or make available nutrients to the plant that the plant absorbs? So root exudation to attract microbes and nutrient cycling versus rhizophagy?
Yeah, so they're not mutually exclusive. Rhizophagy was happening all the time. While people didn't see it happening. It was going on. And so some subset of nutrients that we all assumed is coming out of the soil in soil solution is not coming in soil solution. It's coming in the microbes themselves. It's an overlooked piece of how plants get nutrients. That's the best that I could say. It does differ. It seems like, wow, how could we miss something like this? Well, how could we miss something like that? It's because we have our ideas already clear in our heads about what is going on. And everyone since third grade science, when we're learning plant biology and how roots absorb nutrients into their root hairs from the soil, when we never forget that lesson. And that lesson we carry with us, and it paints everything that we do with plants,
Right.
And so if it's wrong or it's incomplete, we don't know. We can't tell. Now that we know, okay, now the trick is, and it's a very difficult trick, but I mean, we've been trying to do it. It's now separating out, only the nutrients that come in soil solution, only the nutrients that come through that are being oxidized with superoxide, which ones require that system now. But then it's all complexed because what happens when you have the microbes there, they make the roots grow more. And you're going to have, because the roots grow more, you're going to have more microbes there. And your exudation is going to be more. So you're going to have more microbes on the outside too. So it's hard sorting it all out, but you could, in theory at least, design experiments in which you could separate out nutrients absorbed because of growth. And just look at those that are associated with, say, oxidative effects on plants. But it's going to depend on the microbe you use, because they're different microbes and they have different features. So yeah, so sorting it out is not going to be easy, but it could be done. Yeah, so it was always there. Rhizophagy was always happening. It's still happening.
And so some of the rhizophagy nutrient cycling or providing of those nutrients by the endophytes might have been clumped into that passive transport, mass flow. And so do you think that those nutrients that are provided through the rhizophagy cycle, would they, like the nitrogen, the N15, N14, would there be different isotopes that can be tracked, whether it was provided via, soil applied soluble nutrient through passive transport? Or
you could. I actually think that, I mean, now that you raise the issue, Faith, of nitrogen 15, of isotopic nitrogen tracking, what I would like to see, the experiment that I would like to see, would be exposure of trichomes, of leaves with trichomes on them, and to short bursts of 15N nitrogen, right? And then analysis of the 15N that went into the trichome itself versus the hairs. I mean, the hairs themselves, the trichomes themselves versus the leaf itself. And this actually could be done because a plant like hairy mullein, I don't know if you're familiar, Verbascum thapsus, great weed, great weed.
Yeah, I love it.
Does not like nitrogen at all. Does not like it is, but it is filled, the leaves are covered with these branching trichomes that just keep branching like this, just like a network. And so what we could do, the beauty of that is, but we can separate off the trichome layer from the leaf layer just by rubbing it off. And so then we can analyze what goes into the trichome versus what goes in then to the leaf. And then we could track, the movement from the trichome into the leaf like that. I mean, that's the experiment. That's the experiment that we want to do to track this, to track it better. Honestly, the breakthrough came when we were looking at staining bacteria in root hairs to try to figure out what interactions are happening.
Mm-hmm.
Because when we're able to take a chemical equation, you know, and link it to something that the microbe and the plant are doing, you know, things seem more concrete. And then we could just track the chemical stains, you know, through other plant tissues to see how that could happen. So that's what we're doing.
Cool.
But yeah. So anyways, yes.
I'm going to try to get through some of these. We have 10 minutes.
Yeah.
Also, Liz Joseph is hosting a meet and greet for the Soil and Nutrition Conference attendees at I'm at 4:45 my time. Liz, is that going to be 5:45? I know the link is in 4:45. It's in the email that you were sent out earlier today. And it's also on the conference, the Soil and Nutrition Conference website page. And so please join us for a conversation, meet your fellow attendees. And yep, that's Eastern time. Okay, 4:45 Eastern time. Okay, so Tom Wiley asks, a USC Davis soil ecologist told me that though rhizophagy is a real phenomenon, in the case of agronomic crops, that's a selective choice there, its contribution to total nutrient is rather insufficient. Do you have evidence to the contrary?
I don't know how he got that number. Yeah, I don't know where that comes from.
Okay, so Roger Weiss has another question. Roger, thank you for your questions. They're awesome. Are endophytes promiscuous, or is there a specific plant microbe relationship?
Yeah, some of them can go quite a bit between plants and stimulate most of those plants. There are some, though, that when you put them in another plant, they inhibit the plant, and we think it has to do with oxidative susceptibility in the microbe. That, for example, that Micrococcus luteus that I talked about, it produces superoxide, and it will produce some other. It'll produce some other kinds of antioxidants that enables it to resist plant superoxide when it's placed in some other plant. So it can actually inhibit, like, for example, we can take that one from tomato and we can put it in Japanese invasive plant.
Japanese knotweed?
Japanese knotweed, for example. Yeah, put it in that and it inhibits the roots completely. So we use some of those as we use it as a bioherbicidal effects. And so we're actually working on using some of these things, developing some bioherbicides that are kind of endophyte based. And so some of those are used in that kind of a bioherbicide stuff that we're working on. It's really, really cool stuff.
That's a wormhole I would love to.
It's another wormhole. It's another wormhole. We just did a patent on that.
Oh, wow.
Yeah. It's really interesting. Turn endophytes into pathogens and kill the plant. That's the nut on that. That's the whole goal. And so we put a patent out how you do that.
It's like the the insects endopathogenic fungi.
It is now where they're normally symbiotic in there and helpful. But if we provide them arginine, the microbes will take the arginine and they will produce more ethylene. So they'll overproduce ethylene. And then that ethylene
ages the plant.
It ages the plant. It ages the plant, causes stress in the plant, causes plants to die prematurely. So that's the way it is. Yeah.
Wow.
Isn't that cool?
That's so cool.
Isn't that cool?
Oh my goodness. Yeah. And I want to see how it works in nature naturally, because if we're figuring it out, it just means that it's actually happening in nature.
Right.
Okay, so a couple more questions.
Yeah.
Stephen Z says, how much of plant nitrogen needs can be plant nitrogens? Okay, let me start over. How much of a plant's nitrogen needs can a plant get through breaking down the proteins from the microbes it absorbs? All of its needs or a certain percentage?
So the maximum number that I have seen comes from a couple of different experiments, and it's 33% using grasses. Using grasses. Now, do I think that's a maximum? I do not think that's a maximum. I've got a friend who's trying to, Walter Goldstein at Mendowment Institute, some of you may know him, trying to breed corn, organic corn that has nitrogen fixing microbes in it. So we're trying to help him to do that. And we can see the microbes that he's breeding into the corn, and it's having effects on root structure, but it also appears to be increasing root hair trichome length in some of the corn. So we're trying to look closely at that to see if there's any correlation there with his enhanced nitrogen fixation in his plants and like more activity in trichomes.
Every answer you give, I want to ask.
Well, it is this thing. I mean, it goes back to this. We don't provide plants nitrogen in nature. This is what we do in organic, in our production systems. We've made plants now that we put them in systems where we feed them mostly with inorganic nitrogen and in soils that has few microbes in it. And, they don't sustain very well on their own. But if you take, the plants that they came from, the ancestors of those plants, and you look where they grow in nature, they don't need any of that stuff to live. They live perfectly fine on their own without any kind of fertilizers added to them. Plants have their systems. And, if we don't know those systems are there, then we just ignore it, right? We don't know, we do what we're doing. We think our ideas are right about it. And we it's all chemical, it's all chemical. So we just keep putting chemicals on. Okay, but if we have some other things, some other way of thinking about them, so we realize that these are they have their own systems, and then we can start to figure out what those are and figure out, to what benefit, the microbial process really is for plants. But I think that the answer is going to be, it's a fundamental benefit to plants, because if you take away those microbes, those plants do not grow well at all. So it's not a matter, this is not like a casual thing. I mean, we can, I don't think I need to even say anything more about it. Go ahead.
Oh, well, I actually just want to invite you, if you're willing, and no pressure at all. We're having a breakout session after this Soil and Nutrition Conference session. Everyone, if you want to check that out, all the links are in the chat. I personally have a million questions for you, James, but I think that's probably not unusual. And we have many questions here as well. And we have just 2 more minutes, so I could ask a few more questions unless you have something you want to. Okay. Ask. All right. So Lewis asks about the Hackberry experiment. If you've been on the rooftop, then that might be from Matt Power's podcast. Yes. He was wondering what species of Celtis, and do you have any notes, index files, any bibliography for that rooftop experiment for the soilless media? Are you building?
So we did not set that experiment up. Those are all volunteer plants on the roof of this building. And I've been visiting them during the pandemic on a regular basis. So I could see them and monitoring them. And it's amazing. They have no soil at all. These plants, Joe Pye weed is one of them, the Celtis occidentalis, the hackberry, right, occidentalis, Celtis occidentalis. There's a wild, there's some other wild plants that are up there that are like the Thapsus, the hairy mullein that I talked about. It's up there too, growing with no soil. And, you know, those are plants that they obviously, you know, you can't say that they're getting any, at least no applied nitrogen. They're getting, maybe they're getting pollutant forms of nitrogen coming into the air, the dust particles that wash off of the building or whatever, but you can't see any soil accumulation there. They're growing without soil. A lot of plants do that. You know, agaves in the desert, you see them sometimes dangling from a cliffside with hanging by a root, you know, and still doing fine. You know, they're
nature of life to grow.
It is. They will grow. They have their systems. They have their ways, and that's what we have to do. You know, step away from our science for a little bit of everything we know already that we learned from a long time ago, and then start looking with new eyes and see, well, okay, these microbes are in there. Let's see what really they're doing. You know, let's do some experiments. Let's see how much plants really rely on them. You know, stuff like that. You know, so, and that's going to take a lot more than just my lab.
Yes.
Yeah.
Yeah. James, thank you so much for joining us today and sharing all of your information around Rhizophagy cycle, and also sharing the new information around the phylosphere cycle. What will you call that?
I don't know. I haven't. It's not cyclic to me, so I have thought of not calling it a cycle, but it does seem like it could be something important. You know, it does seem important from a number of perspectives. I mean, for one thing, trichomes have these bacteria in them. So, and if they're important sources of other chemicals, then what do these microbes do? I mean, so you could go down the line and find reasons that it's important to look at these trichome microbes, endophytes, a bit more closely. Yeah.
Great. Well, again, open invite if you want to join after this session. And thank you everyone for joining us today. And we look forward to being with you next week, and we'll all just continue building on this knowledge. Have a great day.